Hello. So as Garrett said, initially I studied computer science and biology, and I was going to combine these two in my PhD research. Oh, this is one slide. And I imagined that, computer science would help biology, biology would help computer science, and it would be this nice interdisciplinary collaboration. However, in reality, computer science helps biology a lot, but biology doesn't really help computer science. So is computer science just being a tool here and not really a mutual relationship? For example, if I were to have a list of arithmetic questions and I used a calculator, it's not exactly interdisciplinary math engineering research. And what happens if I had to use my engineering knowledge to build a calculator? It's a little bit more like interdisciplinary collaboration, but it's not quite there. Even so, applying one field as a tool in another field is extremely useful. And so that's really how I'm going to outline my talk. There's various ways of using one field in another field as a tool. You can use one field to develop a conceptual framework in order to better understand a problem. You can use it as a calculator or problem-solving tool. You can use it to generate hypotheses, which you can go back in the lab and test, or you can use it as a communication tool. And I'm using as a case study my own research, which starts off with a biological question, which is, when do genes turn on and off to control every aspect in your body? And I reasoned about it through a physics conceptual framework. Then I simulated the physics using a computer algorithm and used principles from control systems engineering to come up with new hypotheses, which now we're going back to the lab and testing. And, oh dear. And since, okay. And since, okay, that's fine. Also, we needed to come up with a visualization tool so that way biologists would easily be able to use the simulation more effectively. And so let's say this tree is biology. And each leaf is a different specific medical application, like mutation X that's involved in cancer, or signaling pathway Y involved in heart disease. I don't study the leaves. I study the roots, which is the basic science that influences everything. And by this I mean basic principles that exist in all organisms and in every cell, which I think is interesting. And this type of topic is called systems biology. So for an example is, one question I've been thinking about a lot recently is random noise. So sometimes the different processes in your body depend on one or two molecules finding each other. And the time that it takes is really unpredictable. So how can your body control for all these random fluctuations? For example, in these bacteria, all of them have the exact same genetic material. They're all growing on the same plate, yet you can see that they're all different colors. This is because of random fluctuations and noise and just dumb luck. So how can we understand this noise and how biological systems regulate this noise? One of the sources of noise comes from gene regulation. So let's say this is a DNA sequence with a gene and a promoter, which is a regulatory DNA sequence. Proteins called transcription factors bind to the promoter, and based on which transcription factors bind, it causes a different amount of protein to be made. And the protein are the little machines that control everything in your body. However, just because of random fluctuations, you can have different promoter configurations in each of your cells. And each of that can result in different levels of proteins. So the question is, how can we try to predict the distribution of these promoter configurations in cells in your body? And so that's the biological question I was trying to answer. So to think about this, I thought about it from a physics standpoint. And the starting point was based on these really cool experiments where they tracked single molecules under a microscope, and they found out how these transcription factors actually find where they're supposed to stick to the DNA. And they used this process called facilitated diffusion. So facilitated diffusion involves three-dimensional diffusion, which is a really slow process. But it also involves one-dimensional diffusion along the DNA, which dramatically speeds up the process. And the combination of these actually it speeds up the process even more. So this is how, even when you have even 10 transcription factors floating around in your cells, how they can find the right place to bind to turn on a gene. And this is important. We can use this analogy of the theater seating problem. So say you're in an auditorium, and a row in the auditorium is a DNA sequence, and each chair is a place where a transcription factor can bind. So say me and my two friends, Bill and Amy, like to go to the movies, and each of us has our favorite chair. In terms of thermodynamics, the most favorable configuration is everyone sitting in their favorite spot, and then everyone is happiest. And right now, the standard for figuring out where proteins are bound is by looking at the thermodynamics and assuming that everyone will sit in their favorite chair. However, what happens if I'm late coming home from work and instead Bill and Amy arrive first and they sit in their favorite chairs? Now I come along and I have to think, do I want to ask Bill and Amy to stand up and awkwardly move around them? Or do I just want to sit over there and give up? And so because of this, the order of proteins arriving has a huge impact on what the final seating configuration is. And so everyone was using thermodynamics, but we showed that thinking about in terms of diffusion actually creates different and often opposite behaviors. So thinking in terms of physics really helped us understand the certain nuances in the biological system. Now, moving on from physics, now that we have a way of thinking about things in terms of diffusion, and saying, how do we actually calculate this? How do we make predictions using the system? And so as it turns out, just last year, there was a new facilitated diffusion simulation was built. However, it was so slow. Many of the computers in Cambridge are connected into one network, so you can use it as if it were one huge computer. And we were using the more computing power than anyone else in all of Cambridge. Doing a simple simulation would take a weekend. Doing a simulation on a single E. coli cell would take a year. So this wasn't going to work. So we used some computer algorithms to speed up the process. And using computer science knowledge, we were able to decrease the time it would take from weeks or months to seconds. And you could do it on your own little laptop. My MacBook Pro can do what took a year to do on all of the compute power. So that's an example of how using computer algorithms we were able to calculate the solution to the physics problem. And now we wanted to use the simulation to generate hypotheses that we could then go back in the lab and test that would tell us more about the biological system. And to do this, we used a control systems engineering approach. So if you build a computer or something, you try to think in terms of small building blocks. So our building blocks were different combinations of seating arrangements. For example, the switch is where two proteins can bind to the same place. Like, for example, me and Amy share the same favorite seat, so only one of us can sit there at once. The other building blocks we looked at included the barrier, where we have two different transcription factors that like to sit next to each other, or the cluster, where there's two places that can sit that are right next to each other. An example of some of the logic that can arise from these building blocks is the case of the barrier. So the first transcription factor to come in can come in from three different directions, either from coming in from the side or coming in from the top. However, when its neighbor is bound, now it can only come in from two directions. So it's less likely to come in, and it's more likely to come in slower. The third case was the case of the cluster, which is really interesting because on one hand, you have the same effect as before, where if one protein is sitting down, now you can only come in from one side. But in addition, that protein can slide back and forth, which creates all these interesting properties. So each of these building blocks can encode a certain type of logic. If A is sitting down, B can't sit down. If A is sitting down, B is going to come in slower, and so on. And so thinking from a control systems engineer perspective, how can you combine these to create complex logic like a little computer, I guess, thinking about the cell as a computer? So as it turns out, especially in the E. coli genome, there's tons of these really complicated promoters, and each of these creates a specific type of logic pattern. And so we simulated those types of logic patterns. For example, here we look at the cluster switch, which is combining two of those building blocks I mentioned earlier. And then you can come up with some graphical structure that represents logic. And here you can see, in a population of cells, each of these represents a specific promoter configuration, and the thickness of the arrows measures how many transitions between those configurations happen across 20 seconds or so. And you can see that in the case of the cluster switch, the population kind of diverges into these two different populations where either the top transcription factors are bound or the bottom transcription factors are bound. So you create a bistable switch, which you don't see if you combine barriers and switches. So based on this, we can design new configurations and make artificial bacteria, put this artificial DNA sequence into bacteria and see if we can control the system. And that's what we're working on now. In addition, biologists tend to use the software that creates the prettiest graphs. So there's a lot of cases where there's a more mathematically rigorous approach to solving a problem, but everyone uses the one that makes the pretty graphic. So we really, really needed to have a nice way for people to interact with a simulation. And so here's a little website where you can add your data and get pretty interactive graphs that you can click around and explore the data. And so, as you can see from my own research, you can use tools in different ways. And just because you can't have a two-way mutual relationship doesn't mean that you can't have interdisciplinary research. So we created a conceptual framework to understand the problem, where you can see how physics really helps you reason about the biological question. We used computer algorithms to make it possible to solve the physics equations. We used the principles of control systems engineering to come up with a way of generating new hypotheses and entering into synthetic biology. And finally, we used computer programming to create communication tools to allow these ideas to spread. And so the big message is that how can you find another field that can help you in your own work in one of those various ways of applying tools? So thank you.